NK cell work is as follows. So as you may know, MHC class I molecules are expressed on all nucleated cells of a host. So MHC class I on all normal cells, and by normal I mean uninfected cells, is recognized by inhibitory receptors that are on the NK cell. So you can see here that the NK cell has inhibitory receptors that interact with MHC class I molecules, and they also have activating receptors that interact with NK cell activating ligand on the target cell. So NK cells are activated by a balance between signals through the inhibitory receptor and signals through the activating receptor. So for example, if you have more inhibitory signal than activating signal, the NK cell will not be activated and will not kill the target cell. As you can see here, the inhibitory signals that the NK cell is receiving from the MHC class I molecule are inhibiting the activation of the activating receptor such that the NK cell does not kill the normal cell. If you encounter a cell that is missing or that has a different MHC class I molecule than the NK cell can recognize, the cell does not receive that inhibitory signal, and it only receives the positive activating signal through the activating receptor here. This activated NK cell will then release its granule contents into the target cell, leading to the apoptosis of the target cell. In addition, NK cells express the TNF family ligand trail on their cell surface, which can bind and activate DR4 and 5 expressed on some target cells. And this signal will then also activate apoptosis via caspase-9. In viral infections, it's really critical to note this missing or absent MHC class I signal, because many viruses, as we will discuss, will actually knock out or eliminate the expression of MHC class I molecules on their host cell, because these are what are going to activate the adaptive immune response against viruses. So many viruses will eliminate the MHC class I molecule or change it, and this will allow for NK cells to recognize this and to activate the cells. Now, group 1 ILCs are a very weak cytotoxic cells that upon recognition of viruses, the production of IL-12 by viral sensing cells will activate ILC1s, and these cells are very weak cytotoxic cells. And these ILC1s will either kill targets, it's not really known exactly how, but the main thing that they do is produce large amounts of interferon gamma in order to induce activated macrophages. So that is a very general sense of the innate immune responses against viruses. So now that we've gone through the innate, let's dive into the adaptive side, because this holds another key to how we are able to combat viral infections. So first, let's start off with humoral immunity. So humoral immunity to viruses means that B cells will generate antibodies towards the viral pathogens. And this is really critical in two major points. First, opsonization and neutralization of the viruses. Antibody-mediated opsonization of viruses means that viruses that are coated with antibodies to which they are specific will then interact with macrophages or other phagocytosing cells much more strongly, such that antibody-coated virions are phagocytosed and degraded much more readily than viral particles that are not covered in antibodies, which allows for a much greater clearance of the virus after the adaptive immune response is launched against this virus. Antibody-mediated neutralization of viruses is also very critical. So as viruses that are coated with antibodies are being cleared by opsonization, they're not all cleared right away, but that's okay, because when viruses are covered by antibodies, another important thing happens. So typically what happens when a virus is not covered by antibodies is that the virus binds to receptors on the cell surface, and then the virus is brought into the cell. So this is one of the means by which viruses infect host cells. Then the contents are released, and the virus starts replicating in the host. If antibodies cover the virion, this blocks the viral receptors that interact with host cell surface receptors that lead to endocytosis, which means that the viruses that are coated in antibodies, even if they're not opsonized yet, they're uninfectious because they cannot enter host cells. This means that they are neutralized by the antibodies. This is another very, very critical aspect of the humoral response against viruses. Now, linked recognition of antigens by T cells and B cells are really critical in generating the most robust antibody response in the host. Linked recognition is a complex idea, so let's go through it together. So T cells, as you can see here, are activated by antigens that typically reside on the inside of the viral particle. This tends to happen, as you can see here, so the inside of the virus here is in red, the outside is in blue. And you can see that, so with this DC, which is a phagocytosing cell that is also an antigen-presenting cell, they will take up the virus, break it down, and then they'll expose the inner inside of the virus epitopes on MHC class II molecules to epitope-specific CD4 cells. This interaction of the epitope-specific CD4 cells being activated is highly specific to the viral epitope, such that non-specific CD4 cells are not able to be activated. So B cells, or antibodies, they typically recognize cell surface epitopes of virions. So as opposed to the CD8 cells, which recognize the insides, antibodies and B cell receptors tend to recognize the outside. So you can see here the outside of the virus interacting with the B cell receptor. Now, B cell receptors, upon recognition of their antigen, can actually endocytose the pathogen, and this endocytosed molecule is then decomposed and broken down. And then the B cell itself will also act as an antigen-presenting cell and present the inside molecules or inside epitopes of the virus. So here, this provides a very important link between the B cells that are recognizing the outside of the pathogen and the T cells that recognize the inside of the pathogen. And so the fact that the B cells are expressing MHC class I or class II molecules to CD4 cells, as seen here, means that the B cells will actually activate virus-specific TFH cells that provide to help B cells that recognize the linked epitope. And this is really important because TFH cells provide help for producing, opsonizing, and complement fixing antibodies that block viral entry into infected cells. And this is critical because these TFH cells are really the link between the different epitopes that the CD4 cells recognize and the B cells recognize. So we want to make sure that there's linked recognition. And this is how really critical, because if we didn't have a linked recognition system, T cells could recognize one thing, B cells could recognize the other thing, and there's no cross communication. The TFH cells that are virus specific allow for this bridge because they are activated by epitopes that were activated by the external B cell receptor, such that T cells and B cells can coordinate a response to the same pathogen, even though they're recognizing different epitopes of that pathogen. So now that we've started a conversation about T cells, let's dive into the Th1 cells, which are the major class of T cells that are used in an antiviral immune response. So type 1 adaptive immune responses to viruses include CD8 cells, which recognize MHC class I peptide complexes, and CD4 cells, which recognize MHC class II peptide complexes. And both of these naive cells can be activated with the presence of IL-12, and CD4 cells also need interferon gamma to be induced. So Th1 cells are really important. So as I said, they're activated by interferon gamma and IL-12, leading to the transcription of Tbet, leading to their differentiation. And Th1 cells, so these are our CD4 positive cells, are really critical in activating macrophages as they produce high levels of interferon gamma, GM-CSF, TNF-alpha, and CD40 and fast ligands. And they also express IL-3, LT-alpha, and CXCL12, which allow for a more robust immune response against viruses. And as I said, what they majorly do is they produce a lot of TNF-gamma to really activate macrophages and to stimulate their intracellular degradation of intracellular pathogens, be they intracellular bacteria or viruses. So cytotoxic cells are also very critical in the response against intracellular pathogens, including viruses. So cytotoxic T cells will recognize first the pathogen on MHC class I molecules. They will then start proliferating, and will then these effector T cells that are active will kill virus-infected target cells. Now we're going to dive into more about the recognition and effector function of these cells. So CD8 T cells will recognize epitopes that are on MHC class I molecules, seen here. So APCs can actually stimulate effector CD4 cells, and they also stimulate within the same APC CD8 cells. And this is really critical because most CD8 T cell responses are dependent on the presence of CD4 T cells that recognize the same antigen that they do. So what happens is that when an APC interacts with a CD4 cell via an MHC class II molecule seen here, this recognition induces the expression of CD40 ligand, seen here, and IL-2. Now, the stimulation of APC through CD40, so again, that interaction between the CD4 cell and the APC, increases the presence of B7 and 4-1BBL, and these are both really critical to co-stimulate the CD8 cell. So if you can remember, there's multiple signals that are needed to activate an adaptive immune cell. First, we have our the interaction between the MHC class I molecule and the CD8 molecule. We also need co-stimulatory co-stimulatory receptors that also act to activate the CD8 cell, and the CD8 cell also needs the IL-2 produced by the CD4 cell in order to be activated. So this is critical because this is the link between CD4 cells and CD8 cells that recognize the same antigen. Now, the way that CD8 T cells work is once they're activated, they will go from cell to cell, and there will be an initial interaction between CD8 with its target with nonspecific cohesion or adhesion molecules such as LFA1 and ICAM. Once the cells are interacting with adhesion molecules, the CD8 cell, which is just kind of rolling around these cells, if there's no antigen-specific interaction between the CD8 cell and the host cell, then nothing's going to happen, and the cells will separate and the CD8 cell will leave. However, if upon this initial interaction, which brings the two cells in close enough proximity so that the CD8 cell can see if the cell is infected, if once the cell docks onto this potentially infected cell, and if there's this stable pairing of the CD8 molecule to the MHC class I molecule for which it is specific, then the CD8 cell will do a focused release of effector molecules into the target cell, and this target cell will be specifically killed through apoptosis. Now, what's really important to note is that only infected cells are killed by CD8 cells. Again, this is because CD8 cells recognize MHC class I molecules on the surface of the cell, and MHC class I molecules will only express viral epitopes if they are infected. So you can see here that only the center cell is infected, and so it's the only one that has these red virus particles on its MHC class I molecules on its surface. And so this is the only cell that is killed by the CD8 positive T cell, and the neighboring cells are unaffected. So some effector functions. So again, as I've said, the cytotoxic effector molecules that are produced are perforin, granzymes, granulysin, and Fas ligand, which are really critical in killing target cells, as well as some other cytokines that are important in maintaining an immune response. So the way that cells are actually apoptosed is upon engagement of the T cell receptor by the peptide MHC complex, there is going to be a directed release of perforin and granzymes complexed with surgilysin directly into the virally infected cell. So the gran enzyme B is delivered into the cytosol of the infected cell via pores that are made by perforin. So perforin creates those holes through which granzyme B is brought into the cell, and this targets BID and procaspase 3. And truncated BID will then disrupt the outside of the mitochondrial membrane, and then activated caspase 3 will cleave ICAD, which releases caspase-activated DNase. Now, all of these things together, as well as the release of cytochrome C into the cytosol, will activate apoptosis, and CAD will induce DNA fragmentation, another critical aspect of cell death. And it's important that cytotoxic T cells, as well as NK cells, that they induce apoptosis as opposed to other forms of cell death. This is critical because you can imagine if a virus is replicating within the host cell and the cell were just to burst open, you would release all of those viral particles into the host. However, apoptosis is a type of cell death in which the insides of the cell are fully degraded before the cell is removed, and this prevents viral particles from spreading further. Now that we've gone through a very general scheme of how immune responses are generated to viruses, I want to really briefly remind you of the kinetics of antiviral immune responses. So first we have our innate immune response. So we have our production of type 1 interferons, TNF-alpha, and other inflammatory cytokines, followed by group 1 ILCs and NK cell-mediated killing of infectious cells. And only after the innate immune response is unable to maintain or to prevent further infection are T cells and B cells activated in order to generate the adaptive response. Now, you would think that with all of these immune responses, it's kind of crazy that we're ever infected with viruses, right? It seems like every level of viral replication can be counteracted by adaptive immune systems. And this is true. But viruses and other pathogens are very tricky. And they've also evolved mechanisms of evading and subverting the immune responses of our hosts. And this includes different mechanisms, but today I'm only going to go through 3 different types. So first, antigenic drift and shift, subversion of NK cells and cytotoxic T cells, and what latent viruses do. So let's start with antigenic drift and shift, which is a mechanism of viral evolution that single-stranded RNA viruses are very fond of using. So this is an example using very schematically organized influenza virus, which is the major virus that's been studied with antigenic drift and shift. So typically what happens, so this is our influenza virus particle, and you can see that what happens is that on the surface we have antibodies that can bind to this virus, which is great because our B cells generated a very robust reaction against this viral particle. However, viruses evolve very rapidly, and they can actually evolve, they can mutate the epitopes that B cells and T cells recognize such that the neutralizing antibodies can no longer bind those particles. This is antigenic drift. So antigenic drift is when we have an antigen that's on our viral particle, and that virus mutates that antigen or that epitope such that our immune responses can no longer recognize that epitope. Antigenic shift is a little bit different in that RNA viruses also have segmented genomes, which means they have multiple RNA molecules within them. And if two different viral particles, so here we have a red viral particle and a blue viral particle, if they infect the same cell, they can actually shift and trade different mRNA molecules, kind of like cards. And so here you could see that this red virus here, which we have an immune response to, gave away this particular mRNA molecule, which encodes for these receptor interacting molecules here. It switched it with the blue one. And so then when you have a viral immune response, you could see that the viral particle or the antibodies that were generated against this red receptor interacting protein can no longer interact with this one. So antigenic drift and shift work together in viruses in order to prevent hosts from having robust memory responses to different viruses. Now, as you've seen from this lecture, NK cells and cytotoxic T cells are really critical in the immune response against viruses because they eliminate infected host cells. So viruses have evolved a variety of different mechanisms to subvert adaptive immunity and to subvert NK cells and cytotoxic T cells. This is critical as you've seen that adaptive immunity, as well as cytotoxic cells, whether they're innate or adaptive, are really critical in eliminating infected cells and minimizing the spread of infection. There's a variety of different strategies that viruses have evolved to subvert the adaptive immune response. So, for example, they can inhibit humoral immunity by either blocking Fc receptors or making their own Fc receptors so that antibodies, when they bind to infected cells, they can't work. They also can encode different complement factors, a point we didn't talk about today, but that would be really interesting to look at if you would like to check that out in your textbooks. Viruses also have evolved various mechanisms of inhibiting the inflammatory immune response, mostly by using various different cytokine receptor homologs, so that a cell thinks that it's receiving a signal, but it's actually not. The virus has basically exchanged the host receptors for its own, preventing the activation of inflammatory responses, something that we won't go into, but your textbook has more information on this. And what I find to be one of the ways that viruses most effectively evade and subvert the immune response is by blocking antigen presentation and processing. So as you can remember, the mechanism of recognition of infected cells by NK cells or by cytotoxic T cells is the presence of MHC class 1. And so many viruses, including herpes, will actually inhibit MHC class 1 expression so that this prevents cytotoxic T cells from being activated on infected cells. And they can also inhibit peptide transport by TAP to prevent their viral peptides from being associated with MHC class 1. And they can also immunosuppress the host so they can actually generate an anti-inflammatory immune response in the host. So there is immunoevasion of MHC class 1 molecules by viruses. As I mentioned, preventing the expression of MHC class 1 or peptide presentation on MHC class 1 is a very popular method viruses use to prevent an adaptive immune response against them. So you can see here multiple examples of how MHC class 1 molecules are immunoevaded. So for example here, these three viruses can have different proteins that actually block peptides from going into the ER, and this prevents the peptides from being presented on MHC class 1 molecules. These viruses here will actually keep MHC class 1 in the ER and prevent it from going to the surface. And these viruses here will actually lead to the degradation of MHC class 1 molecules in infected cells by various mechanisms. And there are viruses that'll also bind MHC class 1 at the cell surface, so they'll actually bind to it and then prevent CD8 cells from recognizing it. It's not exactly known how this happens or why it works, but it is also a different example of immunoevasion. Here are some examples of immunoevasion. So for example, here we have our viral evasins US6 and ICP47 that block antigen presentation by preventing peptide movement through the TAP peptide transporter. So you can see here that ICP47 is preventing these peptides from going through TAP. And here UCS6 functions with a different mechanism as it's on the cell surface, but again, also preventing peptides from being loaded. Adenovirus protein E19 will actually compete with tapasin. So here is tapasin here that interacts with the MHC class 1 molecule. And so E19 will replace tapasin and prevent peptides from being loaded onto nascent MHC class 1 molecules. And murine gamma herpes virus has an MK3 protein, which is an E3 ubiquitin ligase, which will target MHC class 1 molecules for degradation. So you can see here that MK3 will add ubiquitin to the MHC class 1 molecule, which leads to their degradation. So this is a lot of different mechanisms for preventing MHC class 1 molecules from being expressed or from expressing viral proteins on their surface. So another mechanism of subversion is called latency. So chronic infections are often caused by viruses that are latent. So here, for example, is one of the most infamous viruses that uses latency called Herpes simplex virus. So the initial infection, which happens in the skin, so for example here, the primary infection is typically seen as a cold sore on the lip. This virus is typically cleared at the skin level, but a residual infection will take virions from the skin surface through the nervous system to the trimenial ganglion, and the virus will actually rest there and not replicate and not cause an immune response, and this means that it goes into a latent phase. So the latency phase is a time when there isn't an active infection going on, meaning that the virus is not replicating, but it's kind of being stored away, tucked away in these ganglions, and then you can have a recurrence of infection. This typically happens when your body is really stressed out, if you haven't slept enough, and upon stress, your immune system actually doesn't function as well. So when your immune system isn't as strong, then there's going to be a secondary infection, and this maintains the latency and infection cycle of herpes viruses, and this allows for a chronic infection by the virus. So these were just a few examples of how viruses have evolved to evade and subvert our immune system. So in today's lecture, we've gone through the basics of virology and the Baltimore classification method that allows us to understand different types of viruses and the PAMPs they have. We went through how our innate immune system recognizes our viruses, viral infections, and the first phase of immunity against viruses, including type 1 interferons and type 1 innate immune responses. We went through humoral immunity, Th1 immunity, and cytotoxic immunity towards viruses, and also talked about how viruses have subverted this vast array of immune responses of the host. And that concludes today's lecture on immunity to viruses.